Improvements in technology have allowed us to streamline the poultry industry. In nature, a bird lays an egg in the safety of a nest that will hatch into a young bird. In the poultry industry, we try to mimic the same idea. Under highly regulated conditions, a fertilized chicken egg will be transformed from what we recognize as yolk and egg white into a living, breathing bird. Did you know that not all poultry are the same? In our industry, we focus on 3 types of chickens and 2 types of turkeys. Commercial broiler breeder parent stock produce offspring that will become broilers. Broilers are birds that will be used for chicken meat. Laying hens, however, are birds that lay table eggs, or the eggs we eat. The turkey industry involves turkey breeders, which are the parents of commercial turkeys, and commercial turkeys that are used for turkey meat. Just as not all poultry are the same, neither are all eggs. Only eggs that are fertilized hatch into a chick, although a hen will lay an egg even though it may not have been fertilized. So where do all these birds begin their journey? It happens behind the scenes at a hatchery. Let's take an inside view of the amazing transformation that goes on inside this facility. Eggs that arrive at the hatchery come from breeder farms. Breeders, remember, are the parents of either chickens or turkeys that will be used for meat products or hens that will lay table eggs. Eggs are delivered to the hatchery by specially designed trucks that control the thermal environment to ensure the survival of the embryo inside the egg. These eggs will develop and grow into either a chick, which is a young chicken, or a poult, which is a young turkey, depending on which type of breeder farm they come from. Turkeys are a fairly specialized industry, so we will focus on chicks for this video. Eggs are the unit of life for the developing embryo. Can you see that white spot in the yolk of this egg? This spot is called the germinal disc. If mating has occurred, this is where the sperm fertilizes the egg inside the hen. It is also where the embryo will begin to form. This takes place about 24 hours before the egg is laid. The embryo starts to grow in the female reproductive tract even before a shell is added. Did you ever wonder why an egg has so many parts? The structures in an egg are vital to the survival of the embryo and ultimately a healthy chick. The yolk provides the embryo with nutrients and water. The albumin, or egg white, not only protects the embryo from damage and bacterial infection, but also provides it with a source of protein. The chalazae are twisted, string-like structures that stabilize the yolk in the center of the egg to prevent it from coming in contact against the inside shell wall. If this happened, the embryo could dry out and die. The shell and shell membranes protect the embryo and keep the albumin in the correct position. They allow for gas exchange between the outside and inside environments as an oxygen supply for the embryo. The egg also has an air cell. It is penetrated by the chick's beak during hatching and provides it with its first breath of air. Eggs are placed on trays with the large end up so the air cell is at the top of the egg to ensure proper hatching. Because not all eggs are created equal, it is important that only the highest quality, fertile eggs are selected for incubation. The hatchery wants hatching eggs that are clean, have only one yolk, and are free of cracks and shell defects. In Canada, eggs should weigh a minimum of 52 grams to be put in the incubator. The incubation process involves putting eggs in a controlled environment to allow the embryo to develop properly. Smaller eggs are not set in the incubator, nor are large eggs. Very large eggs often contain 2 yolks and are said to be double yolked. Double yolked eggs will not fit in the incubator trays. If double yolked eggs are set in the incubator, rarely will 1 chick hatch and never will 2 chicks hatch. Hens occasionally lay eggs with flat sides, thin shells, or other deformities. Eggs may become stained, cracked, or soiled on the farm. These eggs are also not set in the incubator, since they may not hatch or they may contaminate other chicks when they do hatch. Breeder producers provide nest boxes for the hens to lay their eggs in a clean environment, but sometimes they will lay eggs on the floor. Eggs that have been laid on the floor at the breeder farm tend to become dirty. Farmers try their best to stop this by encouraging the hens to lay eggs in nest boxes, which are cleaner and make egg collection easier. At breeding farms, eggs are put onto trays with the large end up to keep the air cell in the right location for proper hatching. At the hatchery, eggs are immediately moved from the shipping area to storage. Eggs are stored in a temperature and humidity-controlled storage room until it is time to put them in the incubator. They are kept between 16 and 18 degrees Celsius in storage. The eggs are kept cool to stop the embryo from developing to make sure all the chicks hatch at the same time. Wild birds will purposely avoid sitting on eggs until a certain number have been laid, so that the chicks will hatch at the same time. Eggs are moved onto incubation trolleys. For long-term storage, eggs are turned on the trolleys to maintain albumin quality. Eggs are put in incubators, which provide the optimal environment for the embryo to develop in the egg. Incubators are kept at a temperature of 38 degrees Celsius. The warm air in the incubator starts the embryo's growth again. The eggs spend the next 18 days in this machine, providing enough time for the embryo to develop into a chick. Conditions are kept hot and humid to mimic conditions in nature. Some incubators only house eggs of the same age, so the environment can be specific to the period of growth the chicks are in. This is called single-stage incubation. Other incubators have eggs of different ages. In multi-stage incubation, older eggs are taken out after 18 days when they are finished growing, and younger eggs are put in the other end of the incubator to replace the ones taken out. Older eggs support the development of the younger ones by giving off heat. This is how the eggs move through the multi-stage incubator. This is a candled egg that is ready to go into the incubator. Candling an egg involves shining a light through it to see what is inside. Notice how there is little to see. This egg has been in the incubator for 18 days and is ready to be taken out. The chick will hatch in approximately 3 days. See how the light doesn't shine through where the chick is? The environment in the incubators is key to making sure all the embryos are ready to hatch when they are removed. Eggs are turned every hour or so to make sure the embryo doesn't contact the eggshell, which can lead to dehydration and other problems. In nature, the mother will change the egg's position many times a day. This behavior is part of natural mothering, or brooding behavior as we call it. The embryo develops rapidly inside the egg once incubation begins. After 12 to 13 hours, the primitive streak appears. This is the embryo as it first begins to grow. After 21 to 24 hours, the notochord begins to form, which will become the chick's spine. After 4 days, the allantois is visible. This structure is needed as a place for waste material to collect. Also at this time, the blood supply to the yolk sac is developing, needed for circulating oxygen. After 11 days, the embryo becomes surrounded by the amniotic sac, which protects the embryo. Eggs are taken out of the incubator after 18 days so they can be transferred into hatching trays and placed in the hatcher. Remember, the incubator and hatcher are different chambers with different environments. The incubator trays are removed from the trolley and placed onto the machine. The eggs on the tray are candled with a light, and a computer identifies those eggs that are infertile by measuring how much light passes through the egg. Eggs which have a high amount of light passing through them are infertile. These eggs are picked up by suction cups and discarded. This allows for more room in the hatching tray for chicks to hatch. Next, the remaining eggs are lifted and placed into a hatching tray. Soiled eggs can harbor bacteria, which enter into the shell when the eggs cool. These microbes give off gas so pressure inside the shell increases. Under the right conditions, these eggs may explode, causing the bacteria-infested contents to contaminate the rest of the chicks in the hatching trays. The trays are stacked on a trolley and put into the hatcher. Eggs remain in the hatcher for 3 to 3.5 days, long enough for the chicks to hatch out of their shells. Like in the incubators, temperature and humidity are strictly controlled to ensure optimal conditions for the hatching chicks. Temperature in the hatcher is the same as the incubator. After approximately 3 days, trays of chicks are pulled out of the hatcher. Timing is crucial. The basket must be pulled when most of the chicks hatch. As you can see, this basket is not ready yet. Baskets must be pulled in a timely fashion before the hatched chicks become dehydrated. This basket is ready to be pulled and the chicks processed. Hatch time varies depending on the parent flock age, storage time, egg size, and hatcher conditions. Slower hatching flocks are pulled last. Rising carbon dioxide and decreasing oxygen concentrations signal the chick to begin pipping through the shell. The chick has to first reach its air cell, which is called internal pipping. This is when the chick takes its first breath. The egg can't hold much air, so the chick will then have to break through the shell, which is called external pipping. Chicks turn around inside the egg to crack the shell all the way around. Once other chicks hear pipping, they start to come out of their shells too. A chick that breaks the shell after one rotation will leave a straight crack, which is good. These chicks tend to be stronger and healthier. A badly pipped shell is jagged. The chick probably had a harder time getting out of the shell and took more than one rotation, which takes more energy. Removed baskets of chicks are transported from the hatcher to a separator room. The baskets contain chicks and eggshells which need to be separated before the chicks can be further processed. This helps keep the chicks clean and healthy. A machine gently separates the chicks from the hatch residue, which includes shells and unhatched eggs. Shells are collected in a hopper and are sent for rendering. Unhatched eggs and call chicks are humanely euthanized. Chicks are transported by conveyor belt to the processing room and onto a grading table. Hatching trays are cleaned for reuse. This process is highly efficient because the machine and belts eliminate the need to handle each chick. The area where the chicks are separated from the shells is separate from the processing room for biosecurity reasons. Separation and cleanliness help to keep the chicks healthy. Chicks are visually inspected by workers for navel, down, physical, and nutritional abnormalities. Culls are separated and placed into baskets while good chicks continue on the conveyor belt to be counted. Chicks are culled for a variety of reasons that would prevent their success in a barn, including unhealed navels, poor down, malformations, and nutritional disorders. Healthy birds produce healthy, safe meat products in the long run. Chicks are automatically counted into groups of 100 and placed in plastic boxes. Imagine how long it would take one person to count this many chicks by hand. The boxes are sprayed with a fine mist to vaccinate for certain diseases. The spray lands on the chick's down. The size of the droplets is important for proper coverage of the vaccine. Then the chicks will actually swallow the vaccine as they're grooming or preening themselves, or other chicks. Depending on their destination, the chicks will sometimes have other things done. Birds that require further processing are put on a chick processing table, known as a wheel. Birds are vaccinated, beak trimmed, and toe trimmed on this wheel. Some vaccines are delivered through needle, directly below the loose skin of the neck. When the males and females need to be separated, most of the time, hatchery personnel differentiate sexes in some genetic strains by examining their wing feathers. This method has been used for many years. The females grow their wing feathers faster than the males, so their wing feathers appear longer. This is an example of sex-linked genes being put to use. Keeping everything clean is very important in maintaining the chick's health. HACCP, or Hazard Analysis Critical Control Points, is a program to promote food safety and keeping the industry healthy. Equipment and facilities must be cleaned each day to maximize cleanliness. Biosecurity is maintained all over the hatching facility. This includes hand washing, clothing change, footwear change, and healthy workers. Outside conditions can spell trouble for young chicks and poults. In nature, newly hatched birds are sheltered from the elements by their parents and by a nest. In the same way, the truck that delivers chicks to producers maintains a specific temperature to ensure that chicks survive the trip, especially during cold winter and hot summer days. The truck is outfitted with a power generator to maintain ventilation. For longer trips, chicks are given hatch supplement in the form of a gel puck which contains essential nutrients. With all the activity so soon after hatching, the chicks benefit from eating this source of moisture and food. Chicks are now ready to be delivered to farms, whether it is to laying hen farms that will produce eggs for grocery stores, broiler farms that will produce meat, or to breeder farms to become parents to the rest of the industry. In the case of turkeys, poults will become either breeders or commercial turkeys raised for meat. On a typical day at the hatchery, 100,000 chicks can be hatched and processed and sent out to farms. Though you may not often think about it or see it in action, the hatchery is a very important step in the production of all poultry products.